Alright, so in this presentation, I'll be talking about microplastics in alpine lakes and covering topics such as plastic production, defining microplastics, degradation processes, microplastic morphologies, types of microplastics such as their chemical makeup, pollution pathways, negative effects, looking at one of the first case studies that was published, and talking about some of my own research that I did.
All right, so some background on plastic production. The annual global demand for plastics continues to increase each year and currently stands at about 245 million tons. Being a versatile, lightweight, strong, and potentially transparent material, plastics are ideally suited for a variety of applications. Their low cost, excellent oxygen and moisture barrier properties, bio-inertness, and lightweight material make them excellent packaging material. Alright, so microplastics are defined as plastic debris less than 5 millimeters in diameter formed through various degradation processes of larger plastic debris. They are divided into primary and secondary microplastics based on their sources. So primary microplastics refer to the direct use of microbeads in specific cosmetics and other products which are directly discharged into aquatic environments through human activities. So these don't break down. They're already formed and their size is less than 5 millimeters. And secondary microplastics are plastic fragments formed by larger plastic waste undergoing degradation processes that cause fragmentation and volume reduction. Once plastics enter the environment, external environmental factors such as weathering and solar radiation can break them up, forming microplastics over time. As of today, all the degradation methods for microplastics can be characterized into three categories: physical, chemical, and biological. Physical degradation of a polymer refers to the degradation process of the polymer being exposed to physical conditions such as oxygen, water, pollutants, different temperatures, and mechanical forces, which cause the carbon chain of the polymer to gradually break down and produce small molecules. Chemical degradation refers to the breakdown through chemical processes that alter the molecular structure. This can occur via photooxidation, thermal degradation, chemical oxidation, and biodegradation. The last one is biodegradation, which is the process by which microorganisms such as bacteria and fungi break down organic or synthetic materials into simpler compounds such as carbon dioxide, water, methane, or biomass through enzymatic processes. So once plastics go through one of the degradation processes talked about in the previous slide, they are then classified based upon their morphological characteristics. The most common ones found based off of previous studies and current literature include pellets, foam, film, fragments, and fibers. Fibers and fragments are the two most common found in today's environment, and as you see in the figure on the left, it shows an example of each, and fibers and fragments on the right side. So microplastics are classified based upon their chemical makeup. The most common include polyethylene, which originate from plastic bags, six-pack rings, bottles, netting, straws, milk, and juice jugs. Polypropylene, which originate from rope, bottle caps, and netting. Polystyrene, which originate from plastic utensils and food containers. Foamed polystyrene, which includes floats, bait boxes, and foam cups. Nylon, such as netting and net traps. And polyvinyl chloride, which is plastic film, bottles, and cups. So how do microplastics reach alpine ecosystems? The most common way is through atmospheric deposition. Microplastics can reach these remote and pristine ecosystems even when there are no local point sources of plastic production. Microplastics have been found in remote ecosystems such as the Arctic polar regions, the deep sea environment, and high mountain lakes and glaciers via atmospheric transport and deposition. Due to their low densities, microplastics are often lifted by wind currents into the upper layers of the atmosphere and deposited by wind, snowfall, or rainfall in high mountain ecosystems. So the effects of microplastics is still very limited and still coming out day by day, but so far what's been known is that microplastics in the environment influence soil processes and plant production, alter microbial community composition. They are consumed by biota, leading to impaired health and mortality, transfer up the food chain, and they act as vectors for contaminants. So one of the first publications that came out regarding the presence of microplastics in alpine ecosystems came out in 2022 in the Sierra Nevada National Park in southern Spain. And this study analyzed the abundance of microplastics in the surface of 35 glacial lakes. Water from each lake was sampled using a 250 milliliter jar attached to a 4-meter steel pole. After collection, microplastic sampling was carried out by filtration via glass fiber filters and nitol filters. Both had various pore size openings. The filters were examined using a stereo microscope, and microplastics were then sorted into categories based on their shape, color, nature, and size. The figure to the right shows the 5 basins that were studied in Sierra Nevada National Park. So the morphological results show that fragments were the dominant shape of microplastics, with almost 60% of all particles. Because irregular fragments are usually regarded as secondary microplastics, the large number of fragments indicates that most derived from the degradation of larger plastic. Fibers were then the second most abundant, with 39% of all particles. Based off of previous literature and other studies that have come out recently, fibers and fragments are the 2 most abundant types found today, which align with the results of this study. The figure to the right, part A in the top left, breaks down the shape of the results from each lake. The results based on size show that small particles under 200 microns accounted for more than 60% of all microplastics in this study. The findings are consistent with the predominance of fragments in this study, which under weathering caused by exposure to UV radiation and high temperatures progressively break down into smaller pieces. Particles greater than 500 microns accounted for over 15% of all microplastics, which suggests the possibility of recent contamination, meaning that any particle that was larger than 500 microns could have been recently deposited and hasn't had the time to go through one of the degradation processes, breaking it down into smaller pieces. The results based on color show that more than 50% of the particles were blue. Based off of previous marine microplastic studies, blue is usually the dominant color found in ocean microplastics, as it usually goes unnoticed by birds and fish, which tend to ingest more vivid colors. Also, blue pigments are known to be more resistant to UV degradation compared to other colors. Almost 30% of the microplastics found were transparent, possibly attributing to the contamination by single-use plastic, such as plastic bags, plastic water bottles, etc. If you look at part C of this figure, it breaks down all the microplastics in the study by color. The number of microplastics per liter of water are shown here in figure 3. The full lines and points inside the box represent the median and mean value. The boxes represent the upper and lower quartiles, and the vertical lines indicate the 10th and 90th percentile. The abundance of microplastics in the study using the glass fiber filters ranged from 0.3 to 21.3 microplastics per liter, with a mean concentration that varied from 4.7 with an uncertainty of 3.1 in Dilar to 9.3 with an uncertainty of 7.1 microplastics per liter in Trevelise. In contrast, microplastics using the nitol filters ranged from 0 to 13.9 microplastics per liter in Bulgaria, with a mean concentration that varied from 0 in Lenderhorn to 2.9 with an uncertainty of 5.1 microplastics per liter in Bulgaria. While microplastics less than 50 microns in size represented between 22 and 50% of the total number of plastic particles in the glass fiber filters, these were below 2% in the nitol filters. The comparison between microplastics in these two filters suggested that nitol filters underestimated the less than 50 micron size fraction compared to the glass fiber filters, and therefore may not be a very suitable means to retain and detect the widespread abundance of the smaller size fractions of plastic in aquatic ecosystems. The high elevation of Sierra Nevada lakes in small glacial basins with very limited human activity points to a crucial role of atmospheric transport of microplastic contamination. Once in the troposphere, microplastics are just another type of aerosol that can be transported vast distances and contribute to the flux of microplastics into lakes via wet or dry deposition. Wind patterns of the metropolitan area of Grenada, which is the nearest town to the national park, may carry suspended microplastics and other polluting particles affecting these lakes. The atmospheric transport of microplastics could also be associated with higher frequency and intensity of dust export events from the southern Sahara winds, which is experienced in this region. They also mentioned recreation in national parks has to be taken into account, such as backpacking, camping, and daily visitors, but it is still nearly impossible to quantify the sources of these microplastics that were found. There's been an increasing concern about microplastic pollution, which has led to rapid research growth. The figure provided shows the number of microplastic publications between 1964 and 2022. You can see from 2014 to 2022, there was a dramatic increase in publications. Despite growing awareness and an increase in research, there is still a lack of large-scale, long-term, comprehensive data which alters a complete understanding of the sources, distribution, and impacts of microplastics. Some other problems are the methodologies are still new, and there's accuracy problems with different methods, as you can see with this one in terms of the GFB filters and the Nidal filters. And as of right now, it is still impossible, essentially, to quantify the pollution sources. The next few slides are some research I did this summer after my undergrad program, looking at microplastic contamination in alpine lakes in the Trinity Alps. The ArcGIS map shows the 3 lakes that we sampled. The idea we chose these lakes is that it follows a west to east transect, and we hypothesized 2 pollution sources. Scott Valley on the west is a big agricultural-based economy, and then on the east we have Interstate 5, a highly traveled highway. And we thought that these would be 2 good pollution sources for atmospheric transport of microplastics. And then the 3 pictures show just images of the lakes and their elevation. So this slide outlines my methodology. Go ahead and read through all the bullets. I won't dive into that. But the picture on the left shows our zinc chloride preparation and then our sediment microplastic isolation unit. And so the idea behind the SMI is you put your zinc chloride solution inside of the SMI with a density of 1.7 grams per cubic centimeter, and then you insert 30 to 50 grams of sediment per size fraction, and any microplastic with a density of less than 1.7 grams per cubic centimeter will rise to the top, and anything heavier will sink to the bottom, and most microplastics have a density less than that. You close the tube, and then you pour the top half onto like a glass Wattman filter for stereo microscope identification. My statistical analysis is on a HP computer that's not with me here in San Francisco, but I do have some microscope images of different microplastics that we found throughout the 3 lakes. The majority were fibers and fragments, the pictures I have shown here, which aligns with the current literature themes published right now. It was quite eye-opening to see how contaminated the sediment in these alpine lakes were. And I don't want to throw out numbers of the amount of microplastics, but it was very high compared to what we were hypothesizing.